Good evening, everybody. Welcome to BrisScience for May 2025. It is May, isn't it? Yes. May 2025. Thank you all so much for coming out tonight. If this is your first time here, a special welcome. BrisScience is a series of free public lectures on science brought to you by the University of Queensland, where we aim to bring people who are not just great scientists, but also great communicators to share their passion and their research with Brisbane, and indeed with everybody watching on YouTube at home, because all these talks are being recorded, so you can catch them afterwards. I would like to start by acknowledging the traditional owners of the lands on which we're meeting tonight and pay my respects on behalf of BrisScience to elders past and present. I am your MC for this evening, Joel Gilmore. Before we get started, a little bit of housekeeping, of course. Tonight is going to run, we're going to have a presentation, and I will introduce them shortly. Then there'll be some opportunity for some Q&A, and then we would love for you to hang around for some more questions and some food and drink afterwards. So head out along the side after the talk and hang around and discuss everything you've heard tonight, just like you would after a theatre or a football game or whatever normal people do. Now, questions and answers tonight are going to be done through an app. So look at that beautifully timed QR code there. Thank you to the wonderful tech team here at the State Library of Queensland. If you would like to scan that code, that'll take you to our question page, and you can put in as many questions as you would like, and we'll put that up again after the talk as well, and we will try and get through as many of those as we can after the talk. But while you've got your phones out, great time to put them on silent. Okay, so tonight I am very pleased to introduce Dr Lisette Pregelj from the University of Queensland Biotechnology program. Lisette has a particularly interesting CV with a degree in biotechnology and a PhD from the UQ Business School. And her research interests include the interaction between biotechnology and innovation and regulation. And tonight, she is going to be talking about precision medicine, medicines that are perfectly targeted to the individual, and the progress that we've made over the last 25 years or so. So for a healthy dose of insight with no prescription needed, please welcome Dr. Lisette Pregelj.
Thank you very much, and thank you to the organisers of BrisScience for inviting me here to speak to you tonight. I'm really excited to share part of my research and also part of my journey as a scientist with you all. My name is Dr Lisette Pregelj. I am the Deputy Director of the Biotechnology program at UQ, where I teach biotechnology students all about biology, chemistry, biochemistry, molecular biology, but also the business side of the biotechnology industry and how we can take these amazing new discoveries that are being made in the lab every day onto the market as a product that can help solve some of society's problems. And I've spied a few of my students here tonight. So thank you especially for those who are coming. I'm also a senior lecturer in the School of Chemistry and Molecular Biosciences, where my research focuses on how we can effectively and efficiently develop new innovative drugs and how new innovative drugs and medical products can get to market and how quickly they can get to market to help us and help us more quickly, I suppose. So just before I do get started, just a little disclaimer. So the topic of precision medicine does seem to draw a little bit of thoughts and feelings about medicine, therapy, medical advice. So my presentation is not designed to constitute medical advice. I'm not a medical doctor. As mentioned before, I have a PhD in business, which has nothing to do with medicine. I'm more interested in the industry, the precision medicine industry, and how medicines are developed and trends across that industry. So I will be discussing my own opinions, findings from my research, as well as other scientific papers that have been published. So please don't ask me about a lump or bump on your arm, because I have no idea. I'm not a medical doctor. Before we get too deep into the topic of precision medicine, I just wanted to take a few minutes to talk about precision medicine versus the term personalised medicine, because the terms are often used interchangeably. Some people think they have the same meaning, some people think they have different meanings. I want to explain to you my take on these two terms and where I come from with my research and in my teaching. And that's because this topic of precision medicine or personalised medicine can have a few different meanings. We can think about precision medicine as a verb, the act of doing medicine or treating people in a precise way, or personalising treatment of patients. And many, many doctors will argue that all of their treatment of patients is personalised to the patient that's sitting in front of them. So personalised medicine in that respect is not new and is not innovative because it's been practiced for as long as I suppose we've had medicine. But we can also think about precision medicine and personalised medicine as a noun, as an actual thing, so the medicines themselves. And that's where I come from with my research. I think about precision medicines as the new therapies, innovative therapies that are precise and targeted to individual patients. But there's a little bit of a twist even on that definition, in that what is then necessarily new about having medicines that are precisely prescribed for us? Some would argue that antibiotics are a precision medicine or a personalised medicine because they're prescribed to us based on the type of infection we have, and they're not necessarily new and innovative medicines. So what is it about precision medicine as a noun, the actual medicines, the actual treatments, that is innovative and new, and why I'm interested in them as new technologies and how these new technologies are advancing to the market? So I take the approach that a precision medicine is a medicine that's targeted at patients based on their individual genetic makeup, their individual genomes, their genetic characteristics. So something about your genome, your DNA, that makes you more eligible or more beneficial for certain types of medicines than others. But it could also be the genetics of your disease-causing organisms, so different viruses. So we think about medicines or precision medicines as targeted based on genetic characteristics. This can include drugs targeted for certain tumour profiles, such as an example here, epidermal growth factor receptor mutations, chromosomal abnormalities, such as the homozygous familial hypercholesterolemia, or viral genotypes, such as different virus genotypes. And I'll give a few examples of these types of medicines as we go through the talk. But let's take a step back and actually look at, well, what is a precision medicine? How are they targeted? And why is that important? And why is that beneficial to the patient? So traditionally, and we'll take cancer as an example, majority of precision medicines have been developed and approved to treat cancers. So it's a great example with lots of examples that we can discuss tonight. So if we take a precision cancer medicine as an example, let's look at what that's all about. So traditionally, cancer therapies would have been a one-size-fits-all approach. So every patient who had breast cancer, every patient who had prostate cancer, every patient who had other different kind of groups of cancers were all given the same type of therapy as a one-size-fits-all therapy. But thanks to molecular profiling, thanks to genetic sequencing, thanks to high-throughput screening, thanks to understanding how genes are expressed, being able to visualize that and stain that and actually see how genes are expressed and measured, we're now realizing that there are a whole bunch of subtypes of cancers based on different genetic mutations, such as the BRCA mutation. So BRCA is the breast cancer gene that actually helps prevent breast cancer, and if there's a mutation in that gene, that mechanism can unravel. Other mutations that we're aware of now that characterize different subsets of breast cancer, such as the PLB2 mutations, but not just mutations in particular genes. It could be different ways that the genes are expressed in our cells so that our cells look different or look different from the outside. So different changes in expression of certain genes, such as genes encoding the estrogen receptor, progesterone receptor, human epidermal growth factor receptors. So all these changes in expression, changes in sequences, mutations in genetic sequences, which we are now able to actually understand and measure and test for, means that we are now aware that what we once thought was or breast cancer patients, for example, is actually a whole bunch of different subtypes of patients based on their genetic or genomic characteristics or how their genes are expressed. But it's not just the genes themselves that is impacting precision medicines and how we can improve therapy through precision medicine. We're also seeing a whole bunch of additional tests that we can do to look at, well, what else is happening in our body? How are we metabolizing different drugs? We know that there are different genes encoding different enzymes. That means some of us might metabolize drugs really quickly, so they burn out of our bodies really quickly. Others of us are a bit slower to metabolize those drugs. So these drugs are having different effects on our body based on our own other genetic and genomic characteristics and enzymes that affect drug metabolism. One big one that's being utilised a lot in drug dosing is cytochrome P450s, which is enzymes in the liver that help us metabolize drugs. But there's also a bunch of receptors on our cells that impact how the drugs are taken up into our cells and into our bodies, and also how they're transported around our bodies. So there's all these different expressions and mutations in these genes that don't only affect the type of disease we have, but how we respond to the medication for that disease. So all of this information together means that we can actually now understand that there are different subsets of patients that respond to different drugs differently for different reasons. And we can start to distinguish and define these different groups of patients and develop medicines targeted specifically at those groups based on their genetic and genomic characteristics. A couple of examples, if we think about lung cancer now and lung cancer treatment, and particularly non-small cell lung cancer, is we find that some lung cancer patients have mutations in their EGFR genes, which means that certain drugs don't work for those patients. Instead, they need different types of drugs. And there's three clear examples, lorlatinib, and I'm terrible at pronouncing these drug names. I look at them all day and I read them on my spreadsheet and I read about how they work and I pronounce them in my head and it comes out all gibberish. So please just bear with me when I'm trying to pronounce these drug names. Gefitinib and afatinib. So these are three drugs that have been developed and approved for certain types of lung cancer patients that have this particular EGFR mutation, as opposed to another type of medicines, ALK inhibitors, that have been developed and approved to treat lung cancer patients that have a different mutation. For example, crizotinib, ceritinib, and alectinib. So depending on the patient and if they have a particular mutation, some of these drugs will work for them, some of these drugs won't work for them. But understanding the genetics and genomics of our disease means that we can start to make sure that we're targeting therapies based on what that disease looks like and the subtype of that disease. Now, it's not just cancer. I mentioned that there was a lot of cancer drugs being developed as precision medicines. There's also a lot of work being done in antiviral therapies because there are different genetic subtypes of common viruses that cause disease. So one virus that we have quite a few different treatments for, depending on its genomic subtype, is hepatitis C. So there's actually 8 different variants of hepatitis C, and depending on which genetic variant you have, depends on which drug may or may not work for you. So, for example, Harvoni, which is a drug that's been approved by the US FDA, the Food and Drug Administration in the United States, that was initially approved only to treat genotype 1. So if you had genotype 2, 3, 4, 5, 6, 7, 8, that drug wouldn't work for your infection. So knowing the genetics of your disease-causing organism, in this case hepatitis C, means doctors know which drug to prescribe for that patient, as opposed to genotype 3 infections, which had a different drug developed for it, Daclinza, although that drug has now been discontinued. But the point is that we have different medicines now that target different subtypes of our disease, and we know about this because of all the genetic and genomic information that we're learning. So what are the benefits? Why do we want to subtype disease, understand if there's different genetic mutations causing our diseases, different medicines that might work better for us? Well, I guess that's the answer. It's because the medicines then are tailored specifically to our subtypes of disease, whatever subtype we have. There's no longer a one-size-fits-all approach to treating disease if we understand that there's a genetic component and we understand how different drugs might work for different subsets based on that genetic component. So we can tailor medicine to specific genetic mutations. That then means that that medicine, if it's tailored to us and our genetics and our genomics, it's more likely to work. No longer a one-size-fits-all approach. We're more likely to have a benefit from taking that medicine if it's targeted and tailored. So greater efficacy in the medicine. It also means we might be less likely to have side effects or adverse events or allergic reactions, for example, to the medicine, because again, it's been tailored. That dose has been tailored to how we might be metabolizing drugs. Any mutations that we have or gene variants, that means we metabolize those drugs differently, that we can have our dose adjusted, so we're not having too much or too little medicine. Too much medicine may cause adverse events and side effects, so we don't want to have too much if we don't need it, but then too little, we might not have enough. So once we know about this around our bodies, we can start to use that information in our treatment. So how does this all work? Where has this all kind of come from, I suppose? And how do we know that these medicines are more likely to work or less likely to work? To understand this, it's good to take a step back and think about, well, how are drugs and new drugs traditionally developed and tested? How are they traditionally taken from an invention in the lab onto the market so that we can take them as patients? So that all new drugs undergo through a pretty lengthy, pretty expensive, pretty risky clinical trial process where they're extensively tested through clinical trials. Traditionally, and particularly with non-precision medicines, those clinical trials are what we call blinded or placebo-controlled and randomized. What does that mean? That means that we have a cohort of patients that enroll in a clinical trial, and they are randomly put into two groups, randomized. One of those groups might be lucky to get the treatment. One of those groups may not get the treatment, they may get the placebo. The doctors who are administering the drugs during the clinical trial may not know which group their patient's in, so it could be blinded. And that the patient may not know, the doctor may not know. And it's not until the end of the trial, when they unblind the trial and they say, this group had the new treatment, this group didn't, that they can start to see if there's an effect. So that's what we mean by a placebo-controlled randomized trial. But given that we now know that there are different subsets of disease based on our genetic and genomic characteristics, we might find that the drug that's being tested in this traditional clinical trial design is only effective in a certain subset of the disease. So we're only going to get maybe a couple of people or a small percentage of patients in that trial cohort that's getting the new medicine, the innovative new medicine, that's responding to treatment. We might find we have quite a low response rate, 20%, 40%. In this example, we've had 40% response rate, which is actually really high. Particularly in new cancer clinical trials, you tend to get, and with these traditional trial designs, really low response rate because there's so many differences amongst people because of our genetic and genomic characteristics. The other groups of patients in these trials, even though they're getting the innovative new medicine or new medicine candidate, I should say, it hasn't been approved yet, it's just a candidate, they might have different genetics, different genomics, different mutations, different gene expression patterns that it's affecting how that medicine is working on their disease. So they're not getting a response. So it's all a bit kind of blurry as to, well, how does this medicine work? And what about the people who haven't responded? Does that mean that they can't take this medicine, or what does this all mean? So with precision medicine, they've actually changing the way that we're designing clinical trials to develop and commercialize these new drug candidates, these new medicines. And there's a couple of really, I guess, quite innovative trial designs that are being used to test these new medicines and help us realize and understand the different subsets of patients that respond and don't respond to the new medicines. So one such design is called an umbrella study. So we take all of the patient cohort that are enrolling in a trial, and before the trial starts, we'll do the molecular profiling on those patients to understand what is their genetics, what is their genomics, and how might that affect how they metabolize the drug, how they might react to the drug, how that drug might work, how that treatment might work. We then know going into that trial who is in which group, and we can almost pre-select patients for that trial that we think will respond to that medicine because of their genetics or their genomics in their disease. All of the patients who don't have that marker, that genetic marker, can either be excluded from the trial because we don't think the medicine will work for those disease, that subset of their disease. They could be in a different treatment group, they could be having a placebo, or they could be receiving a non-precision medicine. There is a lot of different details in these trial designs. This is still quite a basic overview. But we don't give them the new medicine because we don't think they're going to respond. This also means that we can reduce clinical trial sizes, which is really important because the bigger a trial, generally the more it costs and the longer it takes. So if we can reduce the size of clinical trials by targeting medicines based on specific subsets of patients and pre-screening based on genetics and genomics, then we can have smaller clinical trials. We also then might see a much higher response rate to that new medicine in that targeted group of patients. So we know then that this new medicine is working better in this group of patients. And we know that this group of patients is similar because of their similar genetics and genomics. That's distinctive from the other group that we've excluded. Also, if the medicine's more likely to work, we can maybe adjust the dose of that medicine down and have smaller doses, which is not only cheaper because you're not paying for as much medicine, but also then might lower side effects. So by targeting patients and thinking about genetics and genomics and how that affects disease, we can see a higher response rate if we're targeting, possibly smaller doses and possibly less side effects. Another option with the umbrella studies is instead of excluding those patients based on their different genes or mutations, we have different treatment groups, and we might test 4 different drugs or 6 different drugs at once, which again is more efficient in terms of new drug development, because instead of testing 1 drug at a time in a trial, we're suddenly testing 6 at once. And we can then start to see if there's different subgroups that respond differently. Another method or another trial design that is being used a lot to test precision medicines is what we call basket studies. And this is where we look at not just specific subsets of a type of cancer, but are there different markers that are consistent across different types of cancers? And does that have an effect on how the drug might work in these patients? And we can then select for patients based on these other particular markers and see if there's an effect across all different types of, for example, cancers, and treat that group. So through these innovative trial designs, and these innovative targeted medicines, we can tailor medicines to specific subsets of disease, which means they're more likely to work and have greater efficacy and less likely to have adverse side effects. This in turn, though, means that it's more quicker and efficient to develop these medicines. And I know this for a fact because I've measured the development time of precision medicines and non-precision medicines. And by that development time, I mean how long were these medicines in clinical trials before they were approved by a drug regulator and able to be sold and marketed to patients. So typically it takes 7 to 8 years to advance through all the clinical trials. There's 3 phases of clinical trials, phase 1, phase 2, and phase 3. They're generally sequential, but you also don't just do 1 phase 1, 1 phase 2, and 1 phase 3 trials. There might be multiple trials at each phase. But it generally takes 7 to 8 years to complete that process. Make sure there's enough evidence to show that the drug's working and it's not providing unwanted risk to patients. New vaccines generally take even longer, up to 15 years. But I studied a cohort of precision medicines that were approved over 5 years, and I found that on average, they only took 5.5 to nearly 6 years to advance through clinical trials. That was a saving of 2 years in clinical trials. That not only was cheaper, because you don't have 2 years of clinical trials, but it meant patients who needed those medicines were getting access to them 2 years sooner, which could be really important depending on the specific disease that the patients had. Just in case you're curious, here's where COVID vaccines sit in relation to this timeline. Mind-blowing. I remember thinking at the beginning, there's no way that a COVID vaccine is going to be approved within 12 months. It takes 5.5 years to approve innovative precision medicines. So yes, we did very well as a society to do that. So just in the second half, I'll just switch focus now to look at, well, where have we come from with our progress of developing these innovative precision medicines? Where have we come from and where are we now in terms of our market and accessibility? So just a bit of a history about where all this has come from. Arguably, the birth of precision medicines was around year 2000, when we first sequenced the human genome and we first gained in-depth understanding of the entire genetic sequence in the human species. And at that time, mapping or sequencing the human genome was predicted to revolutionise the diagnosis, prevention and treatment of most, if not all, human diseases. This was a huge milestone in biotechnology. But that was just the draft sequence, which was published a year later in the journal Nature. It wasn't actually until 2003 that we completed the full sequence. But even then, it took a little bit longer to tidy up some things and finish sequencing part of a final chromosome. I forget which number it was. So it wasn't actually until 2006 that the completed, fully sequenced genome was published in Nature. And some of the scientists were getting a little bit ironic, I suppose, with their titles. Human genome completed, again. We've been promised for 6 years that it had been completed. But turns out that still wasn't quite the case. There were still lots of gaps in our understanding. So work has been ongoing and still is ongoing to fully understand the human genome and our sequence. So I would argue that while 2000 was a great milestone for precision medicine and genomic medicine, we're still making incremental changes and we're still working on that sequence. Even by 2010, so 10 years after the human genome was first, draft sequence was first announced, there was very important scientists who were reflecting back on where we'd come from in 10 years since the human genome was sequenced. For example, Francis Collins, who was actually in the room when Bill Clinton made his announcement about the draft sequence. And on reflection, also published in the journal Nature, it's actually amazing. You can see the entire scientific history by looking through Nature headlines. It's fascinating. So Francis Collins published an opinion piece in the journal Nature that talked about what had happened in the last 10 years and the consequences of that for clinical medicine. And he basically said it had been quite, going the wrong way, and I'm giving away my punchline. Basically, it was quite modest. We've had some advances. New drugs have been developed for some cancers, genetic tests for people with breast cancer, major risk factors for macular degeneration, drug responses predicted for more than a dozen drugs. So 12, 13 drugs maybe. That's pretty good for 10 years worth of work. But he did say that the Human Genome Project has not yet directly affected the healthcare of most individuals. So even 10 years after the Human Genome Project was the draft sequence was first announced, we're still really working to use that data to impact medicine and precision medicine development. And in fact, it wasn't even until 2011 that the term precision medicine was first introduced, and as a distinct concept from personalised medicine, as I mentioned earlier. I personally have been tracking the progress of precision medicines onto the market since 2012. You'll see that a lot of my data does come from the Centre for Drug Evaluation and Research, which is part of the US Food and Drug Administration. That's not because I'm American, I was born in Melbourne. It's because the US market for healthcare is the biggest market globally. It accounts for nearly half of the total market, 50%. It's the kind of first go-to market for innovative new medicines and biotechnology companies generally. And the US FDA is ostensibly the global leader in new drug and innovative drug evaluation and research. So that's a good yardstick by which we can measure progress. So back in 2012, 19% of all of the new innovative medicines that were released on the market by the US FDA were precision medicines. Some examples was Alisio, which is long-term enzyme replacement therapy for adults with the confirmed diagnosis of type 1 Gaucher disease, so a rare genetic disease. Another antiviral for patients that have HIV genotype 1 infection. Another cancer therapeutic for patients that have myelogenous leukemia, but with a specific chromosomal abnormality called the Philadelphia chromosome. So 3 examples of those 19%. Just to compare some of the non-precision medicines approved that year, you can read through those, the indications for which they were approved for. Treatment of advanced renal cell carcinoma, treatment of patients with metastatic castration-resistant prostate cancer, reducing elevated intraocular pressure in patients with glaucoma. There's no mention of genes or mutations or subsets of disease in those non-precision medicines, compared to the precision medicines where we see mention of a rare genetic disease, a subtype of a virus, and a chromosomal abnormality. But what does that 19% actually mean? Is that 19% good or 19% bad? Is that a lot of medicines or a little medicines? So it was actually 7 that year. So 7 precision medicines were approved that year. I guess I'll leave it up to you to decide whether that's a lot or a little. But of those 7, 3 were oncology treatments, 3 were for genetic diseases, and 1 was an antiviral for HIV that I mentioned. Where are we now? So the latest data that I have access to was from 2023, so 1 calendar year out of date, but still gives us a really good idea of where we've come from in the last 12, 13 years. So in 2023, 30% of all medicines approved were precision medicines. Some examples are Gitrio, which is to treat metastatic ROS1 positive non-small cell lung cancer, Afabrio for treatment of adults with Fabry disease, or Serdo to treatment of postmenopausal women or adult men with estrogen receptor positive, HER2 negative, ESR1 mutated advanced or metastatic breast cancer, TrueCap, which is another breast cancer treatment. But we are still seeing lots of non-precision medicines or non-targeted medicines coming through. One for postpartum depression, another depressive disorder. These are just some examples of some of the medicines that have been approved. But again, what is 30%? Is 30% a lot or is 30% a little? It's 16 new drugs. So when you think back to where we were in 2012, that's more than double the number of precision medicines that have been approved. Of those 16, 4 were for oncology, 11 were for genetic diseases, and 1 was for a different type of treatment. But what about in between? Have we had a trend upwards, a trend downwards? So in terms of the numbers of new medicines approved, it sort of stayed fairly consistent for a couple of years, but then really jumped up in the late 2010s, and maybe has settled down a little bit now. We'll have to watch the trend over time. But again, is this a lot or is this a little? When we count them all together, that's actually 144 new precision medicines. Compared to the dozen or 12 that Francis Collins talked about in 2010, I think 144 is a lot. It's a big advancement. Plus, when we think about it in terms of the percentage of new approvals, so we know that 7% back in 2012 was, 7 medicines in 2012 was 20% or 19%. We know that 16 was 30%. The trend has actually been overall increasing over the last decade. So we're actually seeing, on average, about 30% of all of the new medicines that are approved are now precision medicines. And if we look at the trend line over time, it's actually growing. So we're actually getting more and more new medicines that are considered precision medicines. And there's even more that are entering into clinical trials. So we're seeing a really huge growth rate in the number of precision medicine clinical trials that have been registered each year. Huge growth rate in the sort of late 2010s. So I feel like this is great news for the progress of precision medicine onto the market. But that's all US data. What's the situation here in Australia? So here in Australia, our new medicines are regulated by the Therapeutic Goods Administration, and they maintain a database called the Australian Register of Therapeutic Goods. Of those 144 precision medicines that have been approved by the US FDA from 2012 to 2023, 98 of those have been registered in Australia. So they've been reviewed and approved by the Australian Therapeutic Goods Administration. And that's current as of yesterday. And I actually think that's 70% of those medicines are now available in Australia. We need to put this into a little bit of context though. The Australian market does tend to be a little bit slower because we're not as big of a market compared to the US. So some of the drug sponsors don't tend to come to the Australian market because we are quite small, but there's lots of work being done to try and rectify that and encourage all new medicines to be approved, seek approval in Australia. But one good thing about being in Australia is that we have the Pharmaceutical Benefits Scheme. So any medicine that's listed on the Pharmaceutical Benefits Scheme means we get greater access to it because it's reimbursed by the government. And of those 98 medicines that have been registered here in Australia that we have access to, 74, so over 70% of them are listed on the PBS, or they've been recommended to be listed on the PBS and are still kind of working their way through their process. Or they're on what we call the Life Savings Drug Program, which is another reimbursement scheme for drugs that treat really rare diseases. So in terms of progress, I actually think that's really great that we have access to all these great, innovative, new, targeted medicines. So where are we going now? What's next? So we know that we've got diagnosis and treatment for patients based on their subset of diseases, but we're also moving towards prevention. If we understand and know the genetic characteristics of different diseases, maybe we can start to prevent those diseases by early testing for different genetic characteristics. We can also take all the molecular profiling and prognostic markers that we were looking at earlier to understand subsets of diseases and combine them and look at it more holistically. This is actually already happening in terms of what we call a multi-omics approach. The molecular profiling looks at the genomics, the prognostic markers look at the proteomics, so together we call it the omics. But we're also starting to move beyond just genetic and genomic information. We're starting to look at, well, how does patients' medical history affect their response to treatment? How do environmental and social factors affect response to treatment? How do lifestyle factors affect response to treatment? And the elephant in the room, how is AI going to help us sort through and dig through all of these huge amounts of data? Because there's a lot of data here that we need to understand. Lots of different databases that have lifestyle factors, health records, genetic records. How do we bring that all together and understand that and make sense of it all? So in the future, AI will have a big impact in this space, and it's already starting to have an impact. Now, there are some limitations and some drawbacks from this approach. The cost, if we're thinking about targeting medicines to specific subsets of patients, that may be a smaller pool of patients that will actually purchase the drug. it costs a lot of money to develop these new drugs, and the way that the companies recoup that development cost is through sales. And we do see in some rare diseases, and there's programs out there to try and prevent this, but innovation and development stalls because there's no market at the end to sell the drug to pay for the development costs. So costs could become a problem and prohibit development of these drugs, but there are programs in place to avoid that. We are seeing some diseases are hugely complex, and the idea of one gene causing one disease is not necessarily relevant. There's multiple gene variants that could cause multiple diseases. So it's, again, really complex how we work through this data. There's a lot of ethical considerations in linking it back to the costs of development of these drugs. If it's too expensive to develop, well, we still should develop them because people need them. But what if people won't invest in those drugs, and is that ethical or not? Not to mention if we're using AI in drug development and to help speed up and understand links with all this data, integrating all this different data, security of all this different data, and then not to mention at the end of the day, we're talking about human disease, which progresses at the speed of human disease, or as one of my colleagues, Peter Camboris, calls it, the speed of life. So we still may not be able to get drugs onto the market super fast and super cheap because it just takes time for disease to progress so we can test those drugs in patients. But we are starting to make advancements. And I guess the ultimate goal in precision medicine has always been fully precision, almost fully personalised, almost getting back to this term idea of personalised medicine, fully targeted N of 1 treatments. Treatment that is designed specifically for you as an individual or us as individuals. And we are sort of starting to see some of that come to the market. Back in 2019, there was a drug developed just for one person, a little girl called Milla, and they called it Millicen. If you're interested in the story, it's a lovely story. Take a screenshot of this paper and have a Google and have a search of the Millicen story. A drug that was developed for one person and one person only. That's not a solo example. There's another drug that's been developed for a patient called Jackie, called Jacufacin. And that was a mutation that Jackie had in her ALS, or one of her genes causing ALS. But we're actually now seeing that that drug is being developed and it is currently in phase 3 clinical trials, so the last stage of clinical trials before market approval, for 95 patients. And you can actually have a look at a website called clinicaltrials.gov and look at this study and read all about how they're testing this innovative new drug in clinical trials. And there's been so much progress in these targeted N of 1 therapies that the US FDA has even released guidance for developers on how to design their trials and develop these new drugs. And this is really important. The FDA provides a lot of guidance to drug developers, and for them to release a guidance to help drug developers means that they're taking this area seriously. So there has been a lot of progress in precision medicine over the last 25 years, and there's a lot more to come, which I think is great news. So that kind of brings me to the end of the formal part of my talk. I just want to take a few minutes to plug the biotech program at UQ. In case there are any students here in the audience who are interested in studying biotech, please jump on one of these screenshots and learn about the program where you can come and study biotechnology. And actually our program is now 25 years old, and we're having a big celebration this week of biotechnology education at UQ. So please jump on this screenshot if you're interested to hear more about the history of biotech education at UQ. So thank you very much for your time and attention this evening, and I hope you have all learned something.
Amazing. Thank you, Lisette. We'll give you a moment to grab a drink there. We will have some questions now, so we might pop up that QR code again. Hopefully you've all taken note of the 14th of May there for the session on Wednesday, if you're keen. While you are thinking of your questions, we will, of course, be back next month. This is going to be a fun talk. We have Professor Martine Marin talking about the despots of the bird world. Does anyone know which bird we're going to be talking about?
I can think of a few.
Oh, the poor bin chicken. No. It's the adorable honey eater is going to be the topic for next month. To find out why you're going to have to come along. So make sure you sign up to the mailing list, follow us on our various websites and socials, and make sure you get your tickets early because that one definitely will sell out. All right. Are you ready for some questions?
I'm nervous.
We have got a lot of questions here. So, first question, I'm going back here to... okay, Harry asks, is another benefit of precision medicine that the patient does not build up a resistance to a drug which might impact its efficacy?
That's a really interesting question. I don't know the answer to that because I think it would be really drug-specific and disease-specific. So I don't think there's a general rule that we can apply to it. Hopefully, theoretically, I would say yes, that would be a great outcome. But it just depends on the drug and how it works.
Great. Okay, Jonathan would like to know, on a practical level, does the advent of precision medicines mean that medical practitioners will, in the future, screen patients' genetic data for markers prior to prescribing a medicine?
Oh, I don't want to speak for doctors, medical doctors, because I'm not one, but I would think that would be a great outcome because then we can tailor from the beginning what will work and what won't work for you. And you're not taking a medicine that's not going to have any effect and wasting your time. So hopefully, yeah, that would be a great outcome in the future.
Yeah, very important that you're a real doctor, meaning you have a PhD, not one of these. Somebody asked, I'll try and find it, but related here, what does it cost to sequence a genome now? Do you have a sense?
I think it's around $600 and maybe 1 hour. So back in 2000, I think it took 10 years, and I've seen estimates of $300 million to $2 billion. So we've come a long way in the cost of sequencing genomes.
Right. It's impressive technology, Tom. So a couple of questions. Sarah and Bri are interested in CRISPR genetic modification and whether you would call... I think Bri asked, would you include genetic modification to correct defects with CRISPR as a precision medicine? And what are your thoughts on?
Yeah, I personally would because like the example of millicin, that's an antisense oligonucleotide that is blocking expression of a gene that's causing the bad outcome. CRISPR might do something similar and actually go and fix that gene in the first place. There are a lot of ethical issues and debates around CRISPR at the moment in human medicine. I do believe there was a CRISPR-based medicine in trials a few years ago, but it was discontinued. I can't remember the details, but I would think that CRISPR would be a way to go in the future if we can sort through all the ethical issues around using CRISPR. Yeah.
Up for a couple more?
Yep.
Great. We've got a question from Howard. Do we understand how genetics affects disease risk?
Genetics affects disease risk.
I won't put words in your mouth necessarily, Hal, but I'm interpreting this as like, do we understand what is that, why do these markers translate in a certain way?
So for some of the markers, we do know how it translates to increased risk because we can see how the disease progresses based on different markers or different problems. It is hard to make definitive answers. I know there's some studies have been looking at the risk of developing breast cancer if you have the BRCA mutations, but it's based on historical data and observational data. How we can predict in the future, well, I think that's where all of those lifestyle and environmental factors will come into play because it's not just the genes that we have, but it's how they're expressed and then what else is happening in our body, so it's really complex. I think in an ideal world in the future, it would be great if we could have that prediction straight away, but I don't think we're quite there yet.
Yep. A couple of questions from Kevin and Brian around how this might affect various trial processes. In particular, Brian asks, is there corollary? Are there likely to be non-precision drugs which may have unjustifiably failed at the phase 3 trials?
I was actually thinking about that this afternoon. I think if the medicine was being tested in a big enough patient cohort, you would see some effect because there is some subtypes of that disease. There's not, I don't believe there's a huge, oh no, that's not right. There are a lot of drugs that fail phase 3 because of safety, but most drugs do fail phase 3 because of efficacy. What we're actually seeing is almost the reverse positive of that. We're seeing drugs that were approved as a non-precision medicine being targeted and having subsequent approvals because we now know that they actually work better in this subset of patients or less effective in this subset of patients. So we're seeing updates to drugs that were approved as a non-precision medicine initially.
Okay. I've got a question here from Adrian, which is probably a good one just to sort of go right back to basics. So Adrian asks, what is a drug? Chemical, or what are we talking about?
Ooh, I think the definition of a drug is something that has a metabolic effect on the body to treat, diagnose, prevent something disease. There's actually an official definition in the Therapeutic Goods Act, which is part of legislation in Australia. A drug can be a chemical, like a small chemical entity, or it could be a big biological molecule, such as a monoclonal antibody. So different small chemical structures versus large proteins. They all could be considered a drug if they have a therapeutic effect or a preventative effect through metabolic action on our body, is my remembering of the official definitions.
Okay, good. All right, I've got two more. We're going to go for some sort of ethicsy questions now. What is to stop a company from developing a drug for a known rare fatal disease and charging those patients a fortune for it? Ooh. Are there ethics committees in Australia? How do we think about this? Brennan is asking there.
So my understanding is that there is a discussion and a dialogue about what the drug will cost once it gets on the market, particularly if they want it to be covered or if the developer wants it to be covered or reimbursed by the government. There's actually a fair bit of, secrecy around drug prices, but there is a lot of debate and a lot of discussion about reducing drug prices so that they are affordable to patients. What tends to happen in my observation of the industry is that when a new drug is developed and it's launched on the market for an exorbitant price, scientists, doctors, the community gets all up in arms, rightfully so, because that drug is just really expensive. There's a few things that do kind of help prevent that, in that there's special programs to develop drugs for rare diseases, grants, vouchers, ways to collaborate with the regulator through an orphan drug program to try and reduce those costs and get that drug to market quickly to benefit patients, knowing that there's not many people out there that will actually be able to, and not only maybe afford the drug, but not as many patients out there as there, say, is for a general type of cardiac disease. So yeah, there is debate, it is an issue, and it's, I guess, an ongoing debate around scientists. So what's to stop them? Society, I suppose. Us as scientists, us as advocates. Yep.
And important role for everyone to engage, which is, of course, why it's so great to have all of you here at BrisScience tonight to be thinking about these big questions. And there is a related one here from Christine, who asks, how is the health insurance sector adapting to these developments in precision medicine?
Oh, I actually don't know the answer to that, the health insurers. My kind of research focuses more on the development of these new drugs, and then a little bit about how they're reimbursed in Australia through the Pharmaceutical Benefits Scheme. I know that a lot of health insurers, I understand a lot of health insurance companies, some would cover medicine, some don't. It's a different situation in the US. They have a completely different system. But there's been lots of changes in the health insurance sector, I think, that would explain changes in drug coverage more than the types of medicines or the innovations that are coming through. I think it's hard to actually extract the type of medicine from the broader changes in the industry. So yeah.
All right. And I have one last question, which is, you say it was roughly almost 25 years since the genome was sequenced.
Yep.
So if we bring you back in another 20, 25 years for BrisScience, and of course we will still be going in that time, what do you think the big things that, we'll all be amazed at then?
Oh.
Putting you really on the spot here with some long-term thinking.
Do I say what I really think and it's being recorded? Might be a bit controversial.
Definitely. We can totally edit it out.
I think we'll have... I'd like to say we'd have CRISPR medicine. I really like the idea of being able to use CRISPR technology to really solve and prevent disease before we even have it. I don't know. I always think back, what was science fiction 25 years ago that's now a reality? And things like the, mRNA vaccine. When I went through undergrad, that was science fiction, and now we have one. Gene therapy was science fiction, and now it's here. So I guess maybe that's a question for all you guys. What's science fiction now that will be a reality in 25 years?
And what a fantastic call to action to come and get involved in biotechnology, all the things you could discover. So that was an incredible talk. I do have to point out there are a couple of comments that love your shirt as the final thing, just to... so on that note, could you please join me in thanking the incredible Lisette Pregelj. Thank you.
Thank you. And thank you for having me. Oh, thank you.
Please join us for some food and drink, and we'd love to see you all next month. Have a great night.